All right, good morning, everyone. My name is Nate. I'm one of the Medicine Chief Residents. Nice to see everyone in person on Zoom. Today, it's my pleasure to introduce Talia Swartz, MD, PhD, who's an Associate Professor of Medicine and Infectious Diseases and Medical Education at the Icahn School of Medicine at Mount Sinai. She completed her MD, PhD training at Sinai, followed by an internal medicine residency and infectious disease fellowship through the ABIM research pathway at the Mount Sinai Hospital. Dr. Swartz is a physician scientist leading an NIH-funded HIV research program focused on inflammatory signaling that underlies HIV infection. She has mentored more than 80 trainees in her laboratory, published over 50 peer-reviewed articles, and her work has been cited more than 4,500 times. Currently, she serves as a standing member of the NIH National Institute of General Medicine Sciences Workforce Development and Diversity. As the program director of the Medical Scientist Training Program at Mount Sinai, Dr. Swartz has significantly impacted the training and career development of physician scientist trainees. She's a national leader serving as the chair of the Association of American Medical Colleges Group on Research, Education, and Training, MD PhD section, and the executive leadership of the National Association of MD PhD Programs. She is committed to diversifying the physician scientist workforce through efforts to recruit and nurture a diverse group of trainees in the challenging landscape of medicine and biomedical sciences. Dr. Swartz has been recognized for her excellence in teaching and research with numerous awards, including the Junior Faculty Award, Faculty Council Icahn School of Medicine at Mount Sinai, the Excellence in Training Award from the Institute for Medical Education at Mount Sinai. Additionally, she was recognized as a Master Educator from the Institute of Medical Education, the Oswald Avery Award for the Infectious Disease Society of America, and the Terry Ann Kulwich Physician Scientist Award from the Mount Sinai Alumni Association. Please join me in welcoming Dr. Swartz.
Thank you so much, Nate, and good morning, everyone. Thanks so much for coming. I'm going to talk to you today about some of the work that we have been exploring, looking at HIV-driven inflammation, thinking about how we can understand HIV-infected lymphoid tissue from a single-cell analysis. And I hope to convince you that this is an important area of future inquiry and lots of future directions in case any of you is interested in getting involved. I don't have any financial disclosures. So the learning objectives for today are for you to analyze the role of inflammatory signaling in HIV infection and think about the clinical implications for disease progression and comorbidities, and then demonstrate how single-cell technologies can be used to identify and understanding the role of different cell populations in HIV-related inflammation. So I want to start with thanks to all of the people over the years who have contributed to this work. And it's been a huge team effort and a really evolving landscape. And so I'm grateful to each of the people here. So 38 million people worldwide are living with HIV, and this is a huge problem because it has become a chronic illness, and the population living with HIV is aging, that I'll describe in a moment. And we've come such a long way from the early 90s, multiple pills, multiple times a day, very poorly tolerated, to where we are now, where individuals can live for decades suppressed on a single tablet regimen, which is really remarkable and something, the huge testament to the progress of biomedicine in our lifetime. So as you're probably familiar, this is a graph from Tony Fauci from the 90s, looking at the HIV infection timeline. We're looking at the plots of CD4 T cells and the HIV RNA, which are reciprocal. And so during acute infection, there's a drop in CD4 T cells with an associated rise in HIV RNA. And then that can be neutralized due to broadly neutralizing antibodies and host immune responses. And that relates to what can be a decade-long of chronic progression, such that the CD4 T cells will gradually decline with HIV RNA gradually rising until the point of immunocompromise, where the T cells will drop below 200 threshold, HIV RNA will rise, and that's where individuals become at risk for opportunistic infections. Thankfully, this is an older paradigm due to the advent of antiretroviral therapy, where individuals are able to maintain HIV RNA below the limit of detection and a stable level of CD4 T cells. And so this is sort of the state of HIV today, where individuals are, when they have access to ART, able to maintain this chronic infection and really live relatively healthy and long lives. So treatment goals are to reduce HIV-associated morbidity and prolong the duration and quality of survival, which is a reasonable goal, to restore and preserve immunologic function, maximally and durably suppress HIV RNA such that individuals are really below the limit of detection when they come in for visits. And then there are isolated blips that are not uncommon and are not associated with virologic failure. And all of this, all these treatment goals are associated with a reduction or an elimination of HIV transmission. And that's really important so that an individual who is virologically suppressed cannot transmit the virus. So from a public health perspective, that's critical. The problem is that ART is not sufficient for eliminating the virus or the reservoir. And so what we know is that you have active HIV replication, and then when ART is started, you can get the virus below the limit of detection. But the problem is that as soon as we stop antiretroviral therapy, the virus will rebound. And so individuals need to be on lifelong therapy in order to sustain this virologic suppression. And this is a huge barrier to our ability to cure the disease and eliminate HIV as an important cause of morbidity and mortality. So the three main challenges in the field are in these three buckets. This idea of chronic inflammation, that is what's happening with individuals who are on antiretroviral therapy, and we'll talk most about this. Latency, which is what underlies the challenge for cure strategy, that is that the virus can remain latent and then, as we just described, can rebound after cessation of antiretroviral therapy. And then the challenge that the virus is highly mutable, and that is what limits prevention efforts in terms of developing of a vaccine. We're going to focus on the top one here. So as we talk about life expectancy, this is huge in terms of the progress that's been made. So if you look at this top curve, this is the life expectancy for the general population. And then this bottom curve is the life expectancy for people pre-antiretroviral therapy. Thinking about the probability of survival at each of these different ages, and it was really, really dismal. But thanks to improved ART, that curve got shifted upward, and with early ART even further. Early ART is here, improved ART is here, but the important thing to note is that there is still a gap. That is, with optimized antiretroviral therapy, people with HIV are still not living as long as the general population. And so there are a lot of explanations for that. The main one is that people with HIV have multiple comorbidities. That is that if you look at this plot, which looks at the percentage of individuals with 3 or more comorbidities, in the general population, that number increases with age, but it increases hugely in people with HIV. That is that this giant swath of people with HIV have 3 or more comorbidities, and these are associated with the mortality that we see. And these are in multiple categories. There's cardiovascular disease, there's cognitive impairment, there's fractures and osteoporosis, liver disease, kidney disease, and then all of these are associated with poor outcomes. So what's driving these? There's sort of 3 major categories. There's exposures, there's drug effects, that's antiretroviral therapy effects, and then this broad category of inflammation. So exposure, so tobacco and IV drugs are particularly high in the population of people with HIV, and these are independently associated with inflammatory signaling, and we'll talk a little bit about tobacco at the very end of this. In terms of drug effects, a lot of these things, nephrotoxicity, osteoporosis, dyslipidemias, and cardiovascular disease are associated with many of the older drugs, and the newer formulations are less associated with this. A lot of mitochondrial toxicity that are not as much associated with the current more optimized treatment. And then the concept of inflammation, which has a lot of different explanations. The idea that we talked about the limit of detection for HIV RNA, there's the possibility that low-level viremia may potentiate either local or systemic inflammation, reservoirs, and so possibly low level of transmission of virus within tissues where virus is residing may mediate local inflammation. There's abnormal T cell activation, and then compromise of the mucosal integrity that I'll talk more about. And all of this has been described in certain settings being associated with inflammaging, that is inflammatory signaling or chronic inflammation that's associated with premature aging. So if we look at the healthy epithelium of the gut with tight junctions, and you can see the intestinal microbiome is intact through the intact epithelium, but that with HIV infection, the slide is not advancing, yes. So you have denuding of the epithelium, loss of the tight junctions, you have translocation of gut bacteria, and that's associated with lymphocyte homing and innate immune responses. It's associated with IL-6, IL-1 beta, TNF, type 1 interferon secretion. This has been shown in an SIV model in rhesus macaques, but it's well described as a phenomenon that actually results in an immune compromise that is not fully recovered even with full immune reconstitution of the T cell response. And so individuals with HIV then sort of have this happening with a leaky gut, and then that translates to large population outcomes that are associated in people with HIV independently with morbidity and mortality. And these three have come out in multiple trials being associated with mortality, that is hsCRP, IL-6, and D-dimer. And these are all sort of associated with chronic inflammatory states that we're aware of. There have been lots of different clinical trials that have looked at sort of non-specific ways to target inflammation. And none of these have been particularly mechanistic in approach, but have sort of looked to see whether the treatment with any of these drugs might be associated with reduction of some of those inflammatory cytokines, and it has been. But the question really is what are the mechanisms that are underlying this response that is happening when HIV is sort of virologically suppressed? And so one of the things that we've been interested for a long time now is understanding the role of these purinergic receptors. These are purine sensing receptors. So purines like like ATP, which are released from cells, from the mitochondria, when a cell is either dead or dying. So you can see the damaged cell releases ATP, and then that can be recognized by a nearby cell in these three different receptor subtypes. There's P1 receptors that are adenosine receptors, and then P2 types, so P2X and P2Y. P2X receptors are ion channels, and P2Y receptors are G protein coupled receptors. And so what's important about these receptors is that they're known to be upstream of inflammatory signaling, and I will, let me just jump ahead for 1 second. So they're known to be upstream of this inflammatory signaling known as the NLRP3 inflammasome. So you can see that ATP can be sensed here and can then be associated with a large system of inflammatory changes associated with gene expression of the stimulation of pro-inflammatory cytokine transcription, as well as the assembly of the NLRP3 inflammasome, which is associated with a lot of chronic immune conditions and can be associated with the secretion of pro-inflammatory cytokine IL-1 beta and IL-18, and also a lymphocyte-specific form of cell death called pyroptosis. And so we'll come back to this in a minute, but it's important that these upstream receptors have a lot of downstream signaling associated with them. So one of the things that we have been interested in looking at in inhibiting those receptors and downstream to understand the role of that signaling in HIV infection. And I'm going to tell you just broadly about some of the assays that we're using to test this. So we're using a fluorescent HIV reporter virus that is expressing mCherry in place of the viral gene NEF. And so when you look at the cells that are infected in a microscope or by flow cytometry, these cells will fluoresce red. And so in this case, we're using a fluorescent reporter virus that's actually green, similar construct. And what we saw a while back was that if you infect these cells with the fluorescent virus, you get robust infection, a very green population of cells. And when you inhibit with a purinergic receptor antagonist, you almost fully abrogate infection, which is really striking. And we saw that this was the case for different tropisms of virus, and we saw that this was the case in both primary cells and in cell lines. And so the curious question is, so what is the role of these signaling receptors in sort of this downstream inflammation? And is this downstream inflammation and inhibiting associated with some of this chronic inflammation that we see? And so one of the first things that we wanted to ask is this something that we can observe in peripheral blood? And so when we infect peripheral blood with HIV, we do see, like in PBMCs, robust HIV infection, but we don't see secretion of IL-1 beta. And so this has been a phenomenon that's observed by others as well, that there is this sort of cytokine stimulation that's seen, but it's not measurable in peripheral blood. Where it is seen is in lymphoid tissue. And so we began using this lymphoid model where we can either take whole tonsils that are collected from healthy donors undergoing tonsillectomy, and we can dissect these out and cultivate them in a tissue block form, or we can dissociate them into what's called human lymphoid aggregate cultures. And in both cases, we can infect them and observe the course of both infection and the stimulation of pro-inflammatory cytokines over the course of the infection. And then we can also do transcriptomic analysis. And the reason that this tissue is important is that it mimics what's happening in the human state, where the virus is infecting cells, but also where it remains latent. And so we think that it's an important physiological model of what's happening in HIV in lymphoid tissue. Interestingly, when we look at the two different systems, both the HLAC, so the single cell dissociated mixtures versus the tissue blocks, in both cases we're able to see HIV infection. So you can see here, peak infection around day 8, and here we can see cumulative infection over 12 days of infection. But we don't see the stimulation of IL-1 beta secretion in the single cell dissociated mixture. We only see it in the tissue block. And so one of the questions is, so what's the difference? If we just dissociate the cells, we lose the phenomenon of the stimulation of cytokines. And here's a table showing that. So in peripheral blood, we see infection. In both tonsil blocks, we see infection. But in peripheral blood, we don't see the IL-1 beta secretion, and we don't see that in the single cells. We only see that in the tonsil block. So it begs the question of what is unique about this intact tonsil block. So we think it's a bunch of things. We think it's the cell types that are present. We think it's the cell types that are infected. We think it's the cell types that are mediating this close apposition together within this matrix that allow for the stimulation of inflammasome activation, probably, and we'll talk about more, because the cells are close to each other and rely on intracellular communication. So we wanted to ask these questions. So what cell types are present, which cells are infected, and which cells are mediating inflammasome activation? So here's a flow of how we did this. We collected tonsils from healthy donors. We cultivated them on this gel foam collagen raft, and then subjected to single cell isolation, cDNA generation, single cell sequencing, and then are able to perform cluster analysis and then computational analysis to determine which pathways are activated differentially. So first of all, we can see nice infection over the course of 8 days. So what you're looking at here is HIV-exposed tissues with infection over the course of 8 days after infection, and we can maintain pretty good viability over the course of that 8 days. So when we look at what cells are present, the first thing that we want to do is to look at all different tonsil tissues. So we're looking at 7 different donors that are superimposed on each other. And what you can see is that each color represents a different tonsil donor. And there's pretty good overlap on this principal component analysis plot of these different donors, suggesting that there's pretty uniform distribution of different cell types across these different donors. And then when we use cluster analysis using conventional markers, we can see the distribution of all different cell types. And so we can see B cells, we can see T cells, we can see macrophages, squamous cells, et cetera. And when we compress this into more sort of broad categories, we can arrive at these major categories of B cells, T cells, and then dendritic cells, macrophages, and epithelial cells here. But you can see that the majority of cells is T cells. And importantly, these are day 8 cell tonsils, and so This is a snapshot after the course of that infection. And so one of the important questions is, were these the same cells that were there at the beginning and all the way through? Then we can ask the question of which cells are infected, and we use our reporter virus to help us determine this. So what we can do is we can take cells that are sorted for M-cherry positive, those are cells that are infected, and then look at the transcript levels, and that's sort of our positive control. And then we can look at the range of infected cells within our tonsil tissue and arrive at this threshold where everything sort of to the right of this is considered infected. What we see is that in our infected tonsil tissue, we actually have 3 categories of RNA transcript. We have those that are unexposed where we see no transcript. We have the ones that we're calling infected that are positive, M-cherry positive. And then we have ones that are intermediate that have low levels of transcript but do not meet the threshold for M-cherry positive by flow measure. And these cells are an important population of cells that are considered bystanders. These are cells that have some level of virus but are not productively infected. And so we think that the understanding of what's happening in the infected population versus the exposed uninfected is an important distinction that we'll talk a little bit more about. So then when we ask the question of which pathways are most activated in infected cells, these are the pathways that come up as the most significant, and really interesting are these that are associated with oxidative phosphorylation, respiratory electron transport, TCA cycle, complex 1 biogenesis. So all of these are associated with the cell's ability to respire, and it sort of makes sense that a cell that is infected is revved up and has its mitochondrial sort of respiratory electron transport system upregulated. Interestingly, this is not the case for the bystander cells. It's specific to those cells that are infected. And then the other sort of interesting thing that we kind of wondered going into this is, are the same cells that are infected the ones that have that inflammasome activation? And so this here, if you look, this is the HIV transcript. If you look at the cells that have the highest levels of transcript, that's these infected T cells, and then we look at these inflammasome-associated genes, it's not the T cells that have upregulated inflammasome mediators, but it is these cells. It is these macrophages, these exposed, uninfected, or bystander macrophages, which suggests that there's crosstalk, that there's T cells that are infected, and then nearby macrophages that harbor low levels of HIV transcript that are the ones that are activating the inflammasome signaling. And so when we sort of plot by pathway, you can see that again, that the cells that have the highest level of complex 1 biogenesis, respiratory electron transport, or oxidative phosphorylation are those infected T cells. And then the cells that have the highest level of inflammasome activation, so these are 2 pathways that have a lot of those inflammasome gene signatures, it's these bystander macrophages. And so this allowed us to create a model whereby the cells are talking to each other. Infected T cells are talking to bystander macrophages, with pannexin being the missing link. So pannexin is the ATP channel, and what we found was that pannexins are elevated in infected T cells relative to uninfected T cells, and it's these specifically in the T cells that have this elevated oxidative phosphorylation that allows them to sort of rev up their ATP synthesis. Then the ATP goes out through that channel and can signal to these bystander macrophages. And so it allows us to create this model where HIV can infect a CD4 T cell, elevated TCA cycle, elevated respiratory chain. This allows ATP to be effluxed through pannexin and then to be recognized by an exposed macrophage, which can recognize the ATP through its P2X type receptor to activate the inflammasome through sort of that large pathway I showed you before, to result in the synthesis of IL-1 beta and IL-18, which is then associated with this pro-inflammatory signaling. So we thought this was a pretty cool model. And that allows us then to sort of use the single cell approach to look in these human tonsils to identify the fact that most of the cells in this model are lymphocytes, that there are small populations of myeloid cells, but that the CD4 T cells, the ones that have the highest transcript levels, those are the ones that are infected but are signaling to the myeloid cells, which are the ones harboring this intermediate transcript level. These are the bystander cells, and so these are the ones that are responsible for the majority of the inflammatory signaling. And so the infected T cells have activated oxidative phosphorylation, then the bystander myeloid cells have the activated inflammasome signaling pathways, which is a probable reason that the close apposition within the tonsil mediates this communication. So the next question that we wanted to ask is, what about the impact of nicotine? So there's a lot of literature that suggests that nicotine actually interacts with the inflammasome, and there's clearly high rates of nicotine use in people with HIV. And so one of the questions we wanted to ask is there an additive effect or an additional effect of nicotine in tonsils that are infected with HIV? And so very importantly, we know that smokers with HIV lose more years of life from smoking than from HIV. And we know that people who smoke have elevated immune activation beyond that of HIV alone. That is, they have enhanced monocyte activation, and they have enhanced T cell activation. And in both cases, it's really a synergistic effect. It's more than you would expect that's additive for HIV plus nicotine. And so you can see that smokers here don't have a huge difference in the general population, but that smokers here with people with HIV have a huge increase, and the same is true for the monocyte activation. And so when we asked this question, we did a similar analysis to before, where we can now take human tonsils that are either infected or not infected with HIV, and then treated or not treated with nicotine. And so we could do a similar cluster analysis as we did before. We can look at infected versus uninfected cells. For your reference now, if you're remembering the UMaps that I showed you before, the purple lymphocytes were on the right before, so now it's flipped, new set of tonsils. So these are the T cells, these are the B cells, and then we have other smaller populations of myeloid epithelial ILCs here. Again, nice superimposition of different tonsils donors, and then we can sort out the different treatments and exposures of virus. What we see here is that when we look at all these different inflammasome pathways, the thing that is so clear from this is that if we compare nicotine treatment in HIV-exposed tissue or infected tissue, you can see that nicotine treatment does not independently have any effect. And you can see here that HIV exposure does not really have any effect. But that when we have HIV infection here, so HIV infection, you can see that there's some increase, or the yellow relative to blue is upregulation of these pathways. But the thing that should pop out at everybody is that all of these inflammasome pathways are hugely upregulated with HIV infection and nicotine treatment. And so this is really interesting because it's not an additive effect. We don't see this effect with HIV alone or with nicotine alone, but we see this, that there's clearly be a synergy beyond what would be expected. We see this in all cells, we see this in T cells and in myeloid cells. Importantly, in myeloid cells, we don't have HIV infected, we just have HIV exposed, but you can see that in the myeloid cells, it's the exposure of HIV that stimulates this inflammasome activation. So as I mentioned, there's this synergistic effect. So the way that we can calculate synergy is by sort of modeling what we would expect based on the addition of each by itself. So this is just a hypothetical example. If HIV stimulates, like HIV treatment or exposure stimulates to this effect, and nicotine exposure stimulates to this effect, then what we would expect as an additive model is just to add these two together. That would be what we would guess if we added them independently. But then the actual is what happens when we do that experimentally, when we take HIV-infected tissue that's treated with nicotine, and then anything that's sort of above the predicted value would be considered synergistic more than one. And so what we see is that for these NLRP3 pathways, there is a synergistic effect beyond what we would expect, particularly in myeloid cells. That is that here's what we see for HIV alone, here's what we see for nicotine, and then HIV plus nicotine is far and away this this dotted line is what we would expect, and it's way beyond that. And so there are a lot of possible explanations for why that might be. Why might nicotine and HIV have a synergistic effect on this pro-inflammatory signaling? We think that an important link is thioredoxin. So thioredoxin is an important scavenger of reactive oxygen species, and what we observe is that thioredoxin is up in HIV and HIV-infected nicotine treated T cells, and that it's also up in myeloid cells that are exposed to virus treated with nicotine. And so we have a model where HIV infection, similar to what I showed you before, T cells have revved up oxidative phosphorylation with generation of reactive oxygen species that can be recognized in macrophages, nearby myeloid cells, and then this can stimulate IL-1 beta secretion through the inflammasome. But then with nicotine, we see this increased effect where ATP is generated and even more reactive oxygen species are generated, and thioredoxin is even more active to mediate inflammasome activation with even more IL-1 beta secretion. And so we think that it's potentiating all of the effects that we see with HIV alone to an even sort of order of magnitude more. And so as we think about sort of why is this important, we think that targeting these pathways can have powerful impact on sort of as an adjunct to what's currently available in antiretroviral therapy, because what we know is that the drugs are extremely potent in reducing HIV transmission, reducing viral load, but what they're not doing is reducing these comorbidities. And so targeting of the inflammatory signaling will be really important while we are seeking a cure and preventative strategies. A lot of the work that I haven't showed you is collaborative work with Ben Chen and Shram Akbarian, looking at the impact of latency on inflammation in an animal model that maybe we could talk about for next time, but which underlies a lot of the important questions that are limiting our ability to cure the infection, and then prevention efforts, which we won't talk about today, which are critical in developing a preventative strategy sort of on a global scale so that this does not become the problem that it is today. And so with that, I want to thank all of the people that did this work. Ben Chen and Shram Akbarian have collaborative work that we do a lot together, thanks to the Beaumont Lab for a lot of these genomics that I showed you, particularly Nadine Schroed, who did a lot of the computation, Connie Zhao, who is here in the audience, and our funding, and I'm happy to take questions. So thank you so much.